My name is Amina Atiniola with student number 301244531.
And my name is Sirambi Nambadan 3011465997.
And we'll be talking about the effect of antibacterial activity of cinnamon and clove oil against Staphylococcus aureus and Escherichia coli in minced beef. So the first slide, which is the introduction. So meat and all of meat products are naturally susceptible to microbial contamination. And some of the most common spoilage microorganisms are the E. coli and Staphylococcus aureus. So in recent times, refrigeration, which is one of the oldest technique used in extending the shelf life of this product um, has not really been as effective as it used to be, and and synthetic and or artificial products or preservatives has been used in order, and those products have um high health risk. So natural preservatives, which is also known as the plant essential oil, has been used in place of all of these artificial preservatives because they are identified as carcinogenic. So the next slide shows the a picture of the minced meat which we purchased at the food store. So the next slide again is an image of a contaminated minced beef infested with E. coli.
And we can look into the materials and method. And my name is Sarin. These are the materials that we used for the experiment, that is 5 ml TSP for inoculating the organism, and we took 200 gram of minced beef, and we took 3 concentration of both essential oils, the cardamom essential oil and the clove essential oils, and we used 3 concentration 0.5, 1 and 1.5 percentage, and the agar plates are EMB and Bayer Park Agar, and the phosphate buffer is the solution that we used for serial dilution, and we used peptone water for the mixing. And let's look into the inoculation of meat sample and the adding of essential oil. Like first we took 100 gram of beef in a polyethylene bag, and from that we took 25 gram as a control sample, and the rest of the 75 gram was inoculated with E. coli and mixed well for 1 minute. Uh, into that From that we separated 25 gram of inoculated meat sample into 3 polyethylene bags. Into that, 3 concentrations were added into each bags. And these steps were repeated for the S. aureus, but the essential oil was used as the clove essential oil. And we kept these bags at 4 degrees Celsius for incubation. After 3 days, we took the bag and we done the bacteriological analysis. For that, we added 225 ml of peptone water into the each bag and mixed well using a stomacher. From that, we transferred, from each bag, like we transferred 1 ml of a sample into the TSB and incubated at 37 degrees Celsius for 24 hours, and we perform the serial dilution for both the microorganism and kept for incubation at 37 degrees Celsius for 48 hours for S. aureus, and for E. coli we kept at 37 degrees Celsius for 24 hours for E. coli. And this was the serial dilution for the E. coli E. coli with cardamom oil. So we transferred 0.1 ml from each tube, and we incubated the plate at 37 degrees Celsius for 24 hours. And this was the serial dilution for the S. aureus with clove oil. And similarly, we incubated these plates at 37 degrees Celsius for uh, 24 hours.
So the results for the microorganisms after the incubation and after the addition of essential oils at varying concentration of 0.5, 1.0, and 1.5. So for cinnamon oil, we see that it was it was effective at a concentration of 1.5, because we see the total number of colonies of 267 and 127 at plates 10 to the power of 5 and 6 respectively. So the next slide shows the Petri plates after the essential oils were added at the varying concentrations. So the next slide again. Yes, so the next slide shows the results of after the addition of essential oil, which is cinnamon oil, at the same concentration of 0.5, 1.0, and 1.5. But for the 1.5 concentration, we see that it's a TNTC from the results obtained. So it could have been as a result of human error. There was a mislabeling in the plates because the concentration should have been reduced rather than getting these results. So the next slide, please. Thank you. So we see the growth on the Petri plates for S. aureus using the same concentration, which is 0.5, 1.0, and 1.5 respectively.
So while looking into the discussion, the concentration was effective for the E. coli, as seen the results. It like we are possible to decrease the bacterial growth while increasing the concentration. So the essential oil proved to be effective for reducing the bacterial count. But when we are looking for the S. aureus, the bacterial growth was reduced at 0.5 percentage, and at 1.5 percentage concentration it showed a higher amount of bacterial growth. Maybe this is a chance of human error that occurred during the experiment. And from the overall conclusion, um, we can clearly demonstrate the essential oil have the uh, ability to inhibit the growth of a microorganism, and the results are shown that essential oil are associated with greater reduction of bacterial growth. And overall, the experiment provides evidence supporting the antimicrobial activity of essential oils. So it can be seen that the experiment was successful, and essential oil have the ability to reduce the bacterial growth. And this is the reference. And thank you very much. Thank you.
Yeah, thank you so much for listening.